Good afternoon, everyone. I'll be making some forward-looking statements today, so we are a publicly traded company and encourage you to look at our SEC filings for any investment decisions that you may be making. A quick snapshot on the company and who we are. We are a recently formed company, a merger of two companies. We are traded on NASDAQ, and the organization is structured. We have three different facilities, California-based and in New Delhi, and I'll give you some overview of the infrastructure there. Our difference for forming this company, really, we are an autologous-based company, but just like the previous speaker, are very similar to a combination product. We have devices and we have a therapeutic biologic. It's just that the biologic in our situation happens to be minimally manipulated autologous cells. And so under the U.S. definition, those are not considered a biologic product, and therefore we have just been ruled an IDE by the FDA for our phase 3 trial coming up. So I'll talk a little bit about the U.S. structure, but also in Europe we will definitely be a combination product under ATMPs. We are the first commercially viable autologous cell therapy, we think, for the reason that, one, the cells that we're using are safe. The efficacy piece, I'll show you some pretty compelling data in a moment in our human trials to date. The safety factor in our target indications, there have been over 2,000 patients treated in randomized clinical trials globally with autologous bone marrow-derived cells, with over 5-year follow-up. So their safety record is excellent and I think really unmatched in the space. The other interesting factor is we do it all in the operating room, and we do it at the bedside in 60 to 90 minutes. So the patient rolls in, has their bone marrow harvested or aspirated. The particular cells of interest are processed and redelivered through a specific device into the patient, and they roll out the door in about an hour. So that allows us to have a very low-cost delivery process and to do it very quickly so the patient only has an overnight stay in the hospital, if even that. So we think as a company, we're creating significant shareholder value through, one, we've completed 8 pilot human trials, and we've also completed 2 phase 1B clinical trials, and I'll go through some of the data on that in a moment. I think the other issue for our therapies is that they are addressing large target markets, and one, we're focused right now in the cardiovascular space and in the bone marrow transplant and oncology space. We talked a little bit about costing, and I think we've just heard from the payers and other speakers this morning the reason why cost is going to be very important from both showing superiority as well as a cost-effective model. And we believe that a bedside device-based approach with autologous cells is going to deliver the most robust clinical effect at the lowest price point available. So how do we do that in the 60-minute process? Well, we do it through an integrated device platform. Each technology, called our SurgeWorks kit, is the patient comes in. The specific aspiration technology for removing the bone marrow from the iliac crest is used. This is a technology specific to our kit. Cells are quickly removed and they're put into our proprietary VXP smart processing device, which is basically a vision-based system that separates the cells from the patient and prepares an autologous concentrated dose ready for that particular application. Once the dose is prepared, it is quickly checked through an onboard diagnostic system to see that we have the target doses, and we also have them in the therapeutic target range. Because we are dealing with autologous cells, every patient is different. We have to do onboard diagnostics so that we ensure that the purity and the potency of the product is as expected for every patient. And then finally to the delivery. And for each of our indications, each delivery device is uniquely developed so that the target tissue that we're delivering it in, it's delivered in a safe format, and the cells are actually viable as they come out the other end of that needle or catheter or scaffolding. We've done a lot of the preliminary work through our partnership with Fortis Healthcare. To give you a little bit of background on Fortis, they are about 2 times the size of Kaiser Permanente. They are the largest private healthcare provider in Asia. They were the spin-out or the recommissioning of the Ranbaxy family, that former Ranbaxy Pharmaceuticals, and then sold that to Chugai Pharmaceuticals, and they decided to get into the private healthcare market. This gives us access on a daily basis to over 15,000 patients coming through the door. So in our early pilot trials, we can get them into the humans, get it done quickly, find out what works and what doesn't, and reposition ourselves if we need to, or make modifications. To date, we see that that's already saved us in our pilot work about $15 million. So it's a significant cost savings and a way to get products into the market faster that are non-dilutive. Our pipeline overall is very robust. As I mentioned, we are have completed 8 pilot trials. We are moving into a phase 3 trial that we will expect to submit the application this quarter, and then a phase 2 cardiac study. So our two top in the green stoplight in the top-hand corner are our primary targets for this year. The critical limb ischemia for non-revascularizable CLI Rutherford 5 patients, we will be initiating the pivotal trial on, and then for acute myocardial infarction STEMI patients. We've also done about another 600 human patients in the orthopedic and hematology space. I'm not going to address the orthopedic pilot trials today, but several of them have been published, and you can find them on our webpage. Looking at near-term milestones for value creation for our shareholders, as well as pushing our clinical program forward, is, as I mentioned, we will be filing the pivotal trial application this quarter for CLI. We anticipate having the pivotal phase completed by late 2016 and to be commercialized in early 2017. In our bone marrow transplant, and I'll walk through each of these with data in a moment, we anticipate having our 510K for the ABO mismatch submitted in quarter 1, and our haploid product submitted in quarter 3. Looking at the mechanisms, what's happening with our product, and this has been an area of interest, the mechanisms of action are different for each target tissue. So although they're autologous cells injected to different regions of the body do different things. So what are they doing in our target vascular diseases? Well, first and foremost, there's a very strong paracrine effect and the use of endothelial progenitor cells to create new vasculogenesis in the local ischemic region. And so we see 3 main areas of mechanism. One is the implanted cells migrate to the ischemic regions, and they do that very quickly after injection. Then once they've migrated, they start to mobilize the local stem cells in the local tissue, who have started to lose their repair signals from the bone marrow itself. And they also start to send out strong new signals back to the bone marrow to sort of harness a response from the bone marrow niche so that it starts mobilizing new cells to come to that region. So looking at the data and how we progress this, so we started out in animal models, and on the upper left-hand side are the limb ischemia models where you look at injecting the cells, tracking them, and seeing what happens over a 28-day period. What's most interesting about this is that the cells do migrate towards the ischemic region. This is a hind limb mouse model. They do migrate very quickly, but from a safety standpoint, the target cells are actually gone in about 21 days. So within that time period, they've set up a whole new network of bone marrow mobilized cells to continue to do the vasculogenesis. Looking in the middle, the in vitro assays, we prove that, showing that our target cells do form tubules, and then we've translated that into clinical results, very compelling clinical results in CLI patients. Same process we've done in the acute myocardial infarction. We show that in an ischemic heart, the cells migrate through intracoronary injection. They have many mechanisms that they involve in controlling remodeling, as well as filling in the ischemic void area, and then ultimately they end up with volumetric changes and hard clinical endpoint changes at the end, as represented in the MRI figures. So what did we see in the patient once we went through all this process? Well, in our critical limb ischemia phase 1B trial, which was 17 patients, these were all no-option patients. They were near-term amputation, meaning that they had exhausted all of their surgical options and that their only option left was to have their limb amputated right below or above the knee. So we took these patients and we followed them in a 12-month study, and then we followed it up with another additional 12-month study. In the first 12 months, and major amputation-free survival is the main hard primary endpoint that's acceptable for this disease state, at least in the U.S., we had an 82% response rate at the end of 12 months, meaning 82% of these patients who were going to lose their legs in a matter of days to weeks had their leg at the 12-month endpoint. We also just reported about 6 weeks ago, we followed this exact same patient cohort up again, and at 24 months, the same 82.4% still had their legs. So we have a sustainable therapeutic effect in this population. As well, most of these patients, more than 25% of them, would be dead from the cardiovascular event in the first 25 or 12 months, and we didn't see that in the trial either. Our phase 3 we hope to start in the February timeframe. In the AMI model, we're focused on STEMI. We went in for our application for approval to do the pilot trial because we were the first company to come forward with a bedside cell processing system with specific cells of interest and a different delivery device, an intracoronary delivery device. We were allowed to do one patient as a pilot. We took this patient, he was a 43-year-old, had a massive STEMI, was not responding to standard of care 3 days, which is sort of the point where you start to look at prognostics on them, and we did the treatment on him, followed him for 24 months. This guy went from an ejection fraction at 36% to 60%, which is normal for a male of that age within the 24-month endpoint. And we saw the strongest rebound was within the first 6 months. So we're very confident that we are controlling remodeling and we are revascularizing both the heart and the limb, and we think they're the most compelling evidence to date in human trials. Looking at our oncology and hematology programs, we are focused in where our smart vision technology can actually improve bone marrow transplant outcomes. The biggest issue, 2 issues in bone marrow transplant, one is the availability of a matched donor, and the second one is having as many stem cells as possible to transplant into the patient that's had their immune system destroyed through a chemical process. We first look at pediatric patients that have cannot find the right match, and the first issue in pediatric patients, unlike adult patients, is that their red blood cell content has to be very, very low from the donor. As you start to decrease that in the laboratory or minimize the red blood cell contamination, you also simultaneously reduce the stem cells. And with our system, being a smart system, we can actually separate out the red blood cells very effectively while keeping the stem cell dose at a therapeutic or an optimally therapeutic level. We're in the middle now of completing our proof of principle trial, which we hope will take us straight into a 510K for this application because nothing meets the standard in the U.S. now. And we'll be submitting that in the FDA next fiscal year. Second area, which is more regional and really affects our Indian business, is local to Fortis. In India, there is no national marrow donor program, for example, as in the U.S., and there are over 6,000 children that die per year waiting for a bone marrow transplant there that can actually afford to pay for it out of their pocket. What we're doing is we're looking at how we can make the parents, which are only a half match to the child, take the parents' donor cells and make them work in the child, called haploidentical. And we anticipate that this could be over a $150 million market for us alone just in India. This is in partnership with Fortis, and we're about halfway through this clinical trial with very exciting engraftment rates on this methodology. From a corporate structure standpoint, we have a very experienced organization and a very strong management team that have close to 75 years of experience in the space, so we understand how to commercialize cell therapies and devices. And just to sort of wrap up, I think we've outlined that we're safe, Thousands of patients have been treated with these cells before. We ourselves have done over 600. We have shown clinical efficacy in strong phase 1 and 2 studies and are now moving on to our pivotals. And we are addressing large markets of multi-billion-dollar opportunities. The regulatory hurdles we see as being significantly lower to many of our competitors here because getting in phase 1 and phase 2 approval is simpler. Getting through a phase 3 is much more complicated, and this is where safety and efficacy become really, really critical. You show both of them. And then we are cost effective. I don't think we're going to be facing the Gilead issues when we come to the end of the day. These patients, it's going to be a surgical procedure, and it's going to be very affordable. And that means we're going to be reimbursable. So with that, I'll say thank you.